Photosynthesis is one of the most important biological processes on Earth. It is the process by which plants, algae, and some bacteria convert light energy into chemical energy in the form of glucose. This process is essential for life as we know it, as it provides the oxygen we breathe and the food we eat. Without photosynthesis, our planet would be a very different place. Imagine a world without lush forests, vibrant coral reefs, or fields of golden wheat. This is what our planet might look like without photosynthesis. It is the foundation of most ecosystems, providing energy for a vast array of organisms, from tiny bacteria to giant redwood trees. Photosynthesis is the process that sustains life on Earth. Photosynthesis takes place in chloroplasts, which are specialized organelles found in plant cells. Chloroplasts contain chlorophyll, a green pigment that absorbs light energy from the sun. This absorbed light energy is used to power the chemical reactions of photosynthesis. In essence, photosynthesis is like a solar panel, capturing energy from the sun and converting it into a usable form. The process of photosynthesis can be summarized with a simple equation. Carbon dioxide plus water plus light energy equals glucose plus oxygen. This equation represents the elegant dance of molecules that sustains life on our planet. In the following sections, we will delve deeper into the fascinating details of this process, exploring the intricate mechanisms that make life on Earth possible. Chlorophyll is a remarkable molecule. Its green color comes from its ability to absorb light in the red and blue regions of the electromagnetic spectrum while reflecting green light. This is why plants appear green to our eyes. Chlorophyll is found in the thylakoid membranes of chloroplasts, where it acts as a solar panel, capturing photons of light energy from the sun. Imagine chlorophyll molecules as tiny antennas reaching out to capture the sun's rays. When a chlorophyll molecule absorbs a photon of light, it becomes energized, much like a battery being charged. This energized state is short-lived, and the chlorophyll molecule quickly releases the absorbed energy. However, instead of releasing the energy as light, it is transferred to neighboring chlorophyll molecules. This transfer of energy continues until it reaches a special pair of chlorophyll molecules located in a protein complex called the reaction center. Here, the energy is used to excite an electron to a higher energy level. This high-energy electron is then passed along a chain of electron carriers, much like a baton being passed from runner to runner in a relay race. The process of capturing light energy and transferring it to the reaction center is incredibly efficient. It is a testament to the power of natural selection, which has honed this process over billions of years. The energy captured by chlorophyll is the driving force behind the entire photosynthetic process, ultimately leading to the production of glucose. The high-energy electron released from the reaction center chlorophyll molecule begins its journey through an electron transport chain embedded in the thylakoid membrane. As the electron is passed from one carrier molecule to the next, it releases energy. This released energy is used to pump protons or hydrogen ions from the stroma, the fluid-filled space surrounding the thylakoids, into the thylakoid space. This pumping of protons creates a proton gradient across the thylakoid membrane, with a higher concentration of protons inside the thylakoid space than in the stroma. This gradient represents a form of potential energy, much like water stored behind a dam. The potential energy stored in this proton gradient is harnessed by an enzyme complex called ATP synthase. ATP synthase acts like a turbine, allowing protons to flow back down their concentration gradient from the thylakoid space into the stroma. As protons flow through ATP synthase, they cause a rotation of a portion of the enzyme complex. This rotation provides the mechanical energy needed to attach a phosphate group to a molecule of adenosine diphosphate, or ADP, forming adenosine triphosphate, or ATP. ATP is the energy currency of cells, providing the energy needed for a wide variety of cellular processes. The process of generating ATP during photosynthesis is called photophosphorylation. This process effectively converts light energy into chemical energy, storing it in the bonds of ATP molecules. While the light-dependent reactions are busy capturing light energy and converting it into ATP, another crucial process is taking place. Water molecules are split apart in a process called photolysis, releasing electrons, protons, or hydrogen ions, and oxygen as a byproduct. The electrons released during photolysis replace those lost from the reaction center chlorophyll molecules, ensuring that the process can continue. The protons released during photolysis contribute to the proton gradient across the thylakoid membrane, further fueling ATP synthesis. And of course, the oxygen released during photolysis is the oxygen that we breathe, a vital byproduct of this life-sustaining process. Meanwhile, carbon dioxide from the atmosphere enters the chloroplast and diffuses into the stroma, where it will play a key role in the synthesis of glucose. The carbon atoms from carbon dioxide are incorporated into organic molecules in a series of enzyme-catalyzed reactions known as the Calvin cycle. The Calvin cycle is a complex metabolic pathway that takes place in the stroma of the chloroplast. It is during this cycle that the true magic of photosynthesis occurs, the transformation of inorganic carbon dioxide into organic sugar molecules. This process is the essence of photosynthesis, converting light energy into a stable form that can be stored and utilized by all living organisms.